Hello students, myself Abhikumar Sharma, working as assistant professor at GDDSD College, Haryana in the Department of Agriculture. Now today I am going to start with my next topic for BSc Agriculture 6th semester from the course title Plant Pathology. The name of the topic is Role of Toxin in Plant Pathogenesis. The role of toxins in plant pathogenesis. Toxins means harmful substances produced by either host or by the pathogens. Pathogenesis means the manner of development of a disease. In what manner disease developed in a host plant or in a host cell. So let's start with, under this topic we will discuss about phytotoxins, vivotoxins, then pathotoxins, effect of toxins on plant tissue, and at last we will discuss selective and non-selective toxins. So let's start with the definition. What are toxins? Toxins are there are substances produced by a pathogen which is injurious. Injurious means very harmful to host, and it directly and immediately play a crucial means very important role in pathogenesis. Pathogenesis, I already told you, this is the manner of development of a disease. In what manners disease developing inside the host cell. Toxins involved in plant parasites interaction are also called aggressions. So the toxins involved in interactions means how pathogens and host are interacting. Either they are beneficial, either they are harmful on one sided or to the or mutually harm to each other. So this type of interactions, during this type of interaction, the type of toxins are involved are called as aggressions. Now next, according to the germ theory or according to the Koch postulates, a substance to be called a toxins. According to the Koch, which type of substances are called toxins should have the following criterions. The compound must be isolated from the disease plant. A toxin is always produced by a is produced where disease is caused. So the compound which we isolated from the disease plant, it is always toxin, or you can say toxin must be isolated from the disease plant. It should not be present in the healthy plant. So toxins are always harmful, they cause diseases. This means they are always in the unhealthy plant, not in the healthy plant. Compound must be chemically categorized. So we must categorize our compound which and we call it a toxin. What type of chemical composition it have, whether chemically it is poisonous, or at what level or what degree do it have as a poisonous. Third, compound must be next, fourth, when isolated toxin is reintroduced to a healthy host, it should produce the original symptoms. Or if after isolating it from the unhealthy plant and injected it to the healthy plant, it always produce original symptom that were in the unhealthy plant. So before going further, first of all we will see, here we having a structure of a plant cell. There are different target sites at where the pathogens attacks or different type of toxins are produced. So let's see here in the outside, you are seeing there is a cell wall. As we all know ki plant cell contain cell wall, and inside the cell wall presence of there is a presence of cell membrane, and in the center there is a large vacuole that is responsible for the regulation of the cell. Inside there is a nucleus, and outside the nucleus there is a nuclear membrane, and beyond the nuclear membrane in the cytoplasm there is a endoplasmic reticulum that is mainly used as a it is of two type, rough and smooth. Rough is mainly consist of ribosome that are the main site for the protein synthesis, and smooth are for the synthesis of lipids. Then next, mitochondria, it is the powerhouse of the cell. Then chloroplast, it is coming under the organelle plastid, as it contain a pigment that is chlorophyll. That's why we call this organelle is a chloroplast. Golgi apparatus, they are used for packaging of different type of material within the cytoplasm, or for the transportation from in or out of the cell. Then these all are the different sites where interactions of this host cell with pathogens takes place. So next, we are classifying the toxins as per our syllabus according to the source of origin from where they are originated. Either it is produced by host or by pathogen, or by after the interaction of both host and pathogens. We divided the toxins into three types: phytotoxins, then first phyto, patho, and vivotoxins. So first of all, we will discuss about pathotoxins. As the name suggests, patho means by the pathogens. The toxins are mainly produced by the pathogen. These are the toxins which play a major role in disease production. These toxins are very full, having a very crucial role in the production of disease, and produce all or most of the symptoms characteristics of the disease in the susceptible plant. Susceptible plants are those plants that are suffering from disease, or they can easily acquire a disease wi- with a short attack or with a limited attack of the pathogens. So susceptible plants are always showing symptoms that they are suffering from different kind of tissue damaging or necrosis of the cells or different types of disease. So most of these toxins are produced by pathogen during pathogenesis. So these toxins are produced when the after the attack of pathogens, and during pathogenesis, that during the development of the disease, the pathogens producing toxins. Thus different examples of uh, pathotoxins are Victorian, that is produced by the pathogen Colicobolus victoria, and Helminthisosporium victoriae. The causal agents of Victoria blight of oats, this is the host specific toxin, that is in a particular host, that as we seen in the example, that in the host only these pathogen will produce this toxin, that is Victorian. Next, we are discussing phytotoxins. These are the substances produced in the host plant due to host pathogen interaction. These types of the toxins are produced after the attack of the pathogens, when pathogen interacting with the host plant. At that time, phytotoxins are produced, for which the causal role of disease is merely suspected rather than established. So the role of disease, the disease is merely suspected. So we suspect, this means suspect means करना होता है शक करना. So we think so कि if toxins are produced, then disease plant is suffering, or host plant cell is suffering with a disease. These are the products of parasites. How the phytotoxins are the mainly the final product of parasites, which induce, when these toxins are produced, when parasite induce few or none of the symptoms caused by the living pathogens. So here pathogens, after interacting with the host cell, produce some symptoms. This will produce phytotoxins. They are non-specific. Even non-specific mean this means they are not host specific. Even after dispersal, or non-susceptible plant are also get harmed with this type of toxins. They are non-specific, and they are no relationship between toxin production and pathogenicity of disease causing agent. So there is no relationship, there is no uh, interaction. There is no any passage of disease between toxin production and pathogenicity. After pathogenicity, before and during pathogenicity, at any time toxins can be produced. The example of phytotoxins are alternaric acid that is produced by Alternaria solani, and pericularin toxins produced by Pyricularia oryzae. Third one is the vivotoxins. These are the substances produced in the infected host. These are also substances, this means harmful substances. These are the all toxins. These are produced in the host by the pathogens. When pathogens attacked within the host during its pathogenicity, pathogen produce a toxins, or it its host which function in the production of the disease but is not itself the initial inciting agent of the disease. Or even these vivotoxins are either produced by the pathogens and also by the host, but the difference is that itself the initial inciting agent of the disease, but host not a initiator. After the attack of the pathogen only it produces vivotoxins. Example is fusaric acid, which cause wilt causing, the pathogen is wilt causing fusarium species which cause wilting in the plants. Now next is toxin in plant parasite interaction. After the interaction of plant with the parasite, that is pathogens, two types of toxins are produced. One is general toxin and second is specific toxin. General toxin means non-host specific or non-host selective toxins. Specific toxins are host specific and host selective toxin. General toxins either they produce in within the cell or in the host, not within the limits. Beyond the limits it also produce over the non-susceptible plants also, but specific toxins are always produced within the limits or within their at their particular host plant or a cell. So you can see here the examples of journals or non-host specific toxins are tab toxin, physiolotoxins, ten toxins, or circosporin. And here the specific toxins are Victorian, T toxin, HC toxin, and AM toxins. Later we will further discuss about the whole description about these toxins. Let's how toxin in plant pathogens interaction are classified. As we have already discussed in the previous table, once again I'll told you. Toxins in plant parasite interaction are classified into two groups, that is general toxin and specific toxin. General toxin are non-host or non-specific, and specific toxins are host specific or host selective toxins. So let's start with general toxin. They are also called non-host specific toxins or non-host selective toxins. These toxins produce all or part of the disease syndrome, or not only on the host plant but also on the other species of plant that are not normally attacked by the pathogens in nature. These toxins produce symptoms within the disease plant as well as over the healthy plants also. This means that general toxins are not host specific or host selective, as we already discussed. Specific pathogens can produce these toxins in any host species. So specific pathogens can produce toxin in any host, not in a particular host. They are viable, they can produce toxin in any kind of species or any kind of host plant. Next, the examples of different type of non-host specific toxins are, you can see here different examples. And the first is tab toxin, then toxin, then ten toxin or circosporin. So first of all, we discuss about the tab toxin. This is the structure of the tab toxins, where it is basically a protein made up of number of amino acids. You can see here there is the formation of peptide bonds between amine and carboxyl group. The wildfire toxins, that is tab toxins. Wildfire toxins is generally toxins produced by bacterium Pseudomonas syringae and the species are tabaki. This pathogen causes wildfire diseases in tobacco. Isolated toxins produce similar disease in other plant also. That we all know about, as we discussed, these are non-host specific. So this toxin can produce disease in all type of plants, in disease as well as other plant also produce similar type of symptoms. Toxin is a dipeptide toxin, that is dipeptide made up of two amino acids. After joining of two amino acids with the help of peptide bond, make peptide bond composed of two amino acids, that is threonine and tabozinin. Tabotoxin as such is not a toxin to plant. In the cell it become hydrolyzed to release tabozinin, and tabozinin inactivates the enzyme glutamine synthase in plants. Inactivation of glutamine synthase lead to the depleted glutamine level. This result in the accumulation of ammonia in the cell. So ultimately, after the accumulation of ammonia within the cell, which leads to the cell death and causes different types of symptoms within the host cell. Second is physiotoxins. Physiotoxins is generally toxin produced by the bacterium. This toxin is also produced by the bacterium Pseudomonas pharyngeae physiolocola. The structure of physiotoxin you can see here. This bacterium causes halo blight in beans and other legumes. This toxin is mainly produced within the legumes plants. The plant treated with this toxin show original symptoms on non-host plants, reduced growth. These toxins ultimately produce its effect by reducing the growth rate of the plant, disruption of apical dominance, that is no apical bud formation or no differentiation of apical part of the plant. Accumulation of amino acid or ethanoline are the effect of the physiotoxins. Physiotoxins is tripeptide. Here three amino acids are included for the formation in the chemical composition of physiotoxin, that is orethionine, alanine, arginine. This tripeptide also carries a sulfamyl group. This group is mainly responsible for the functional amity of the toxins. The toxin is cleaved by the host enzyme to release alanine, arginine, and phosphosulfamyloorethionein. And this PSMO is the functional amity of the toxin. Next is the third type of non-host specific toxins are teno- tentoxin. Tentoxin is generally, you can see here, it is a general toxin produced by the fungus. This toxin is mainly produced by the name of the fungus is Alternaria alternata. Alternaria alternata causes chlorosis in seedling of many plants. Chlorosis means the depletion of chlorophyll from the leaf part of the cells, which causes yellowing of leaves. And ultimately Alternaria alternata causes chlorosis in seedling of many plants. Toxin is a cyclic tetrapeptide. Cyclic tetramer, it is in the cyclic form, and tetrapeptide, there are four types of amino acids joined to form tentoxin. Tentoxin binds and inactivates protein involved in the energy transfer in chloroplast. It also inhibits the phosphorylation process in the leaves and thus reduce the formation of ATP. So ATP is it is the energy source in the plant or in the most in all organisms, that is adenosine triphosphate. If ATP is not produced, then further biochemical processes are not able to take place. So next, 10 toxin is also interfere with normal chloroplast development and chlorophyll synthesis and thus induce chlorosis in leaf. As I already told, chlorosis means yellowing of leaf when there is a no proper development of chloroplast. So then there will be a there will be no photosynthesis or no formation of green leaves takes place, or existing leaves become decolorized, that is their color get faint, or leaf get changes into yellow color. The exact mechanism of action of the 10 toxin is still not clear. Then fourth one, last non-host specific, is cercosporin. Cercosporin is a general fungal toxin produced by many species of Cercospora. This pathogen causes leaf spots and leaf blight disease in several plants. The cercosporin toxin is activated by the light. As the light fall over the plants, this toxin get activated and produce different type of diseases like spots over the leaves and leaf blight and disease, which ultimately reduce the growth of the plant, and plant have to be suffered with different type of symptoms. Ultimately, even the death of the plant takes place. Activated toxin react with oxygen to produce singlet oxygen, that is nascent oxygen, single atom of oxygen. This reactive singlet oxygen destroy the host membrane system. Host membrane, that is plasma membrane, are get destroyed with after reacting of singlet oxygen with the membrane system, and they can react with lipids, proteins and nucleic acids of the host cell. This toxins, cercosporin, react with lipids, proteins and nucleic acids of the host cell. Ultimately, the fungal mycelium is able to tolerate the toxins. The fungal mycelium produce pyridozin, vitamin B6, which neutralize the reactive oxygen. Then second type of toxins, that are host specific or specific toxins. The characteristics of host specific toxins are, they are also called the host specific toxin and host selective toxins. These toxins produce the disease symptoms not only on specific parasite and host combinations, particular that they act over a particular host only. Specific toxins are not produced when pathogen attack a non-host plant or known or another host plant. They are always host specific, so there is no attack of these toxins over the non-host plants. Then this means the specific toxins are host specific and host selective toxins. Specific pathogens can produce these toxins only in specific host species, as already discussed. So what are the examples? And you can see here the four examples we having with us for the host specific toxin. The first toxin, host specific toxin, is Victorian. Then second, T toxins, and third, HC toxin, and fourth, AM toxins. So first of all, we will discuss about the Victorium. You can see here the structure of Victorium. Then this toxin is produced by Helminthosporium victoria, and this pathogen is very selective and specifically infect the Victoria varieties of oats. Only Victoria varieties of oats are affected with this pathogens. Pathogens infect the basal portion of the host and produce toxins. This pathogen may mainly attack over the basal part of the hosts, and this toxin then transcolated the leaves where it causes the blight disease. This toxin ultimately from the basal part translocate with the help of, you can, with the help of phloem or to the leaf part, where it produce leaf blight of blight diseases in the leaves. In severe infection, the plant is destroyed completely, even it cause the death of the whole plant. The pathogens is not able to produce the Victorium toxins when they are infected, when they infect other species or even other varieties of oats. Toxin produced in the fungus is controlled by a single gene. This toxin produced by the fungus is controlled by a single gene. Second type of host specific toxin is T toxin, produced by T race of Coccolobolus, that is Helminthosporium heterostrophus. This pathogen causes leaf blight in corns, then last toxin, that is Victorian, was specific for the oats plants. In this plant mainly causes diseases. These toxins mainly cause disease in corns. Tea rays differ from the other strains of the pathogen only in the ability to produce these toxins. T rays or T toxins only attack the Texas male sterile cultivars of cones only, as we discussed in the plant breeding. Male sterility, that is non-functioning of male part of the flower. So this toxin is responsible for the male sterility in the cultivars of cones, which make the non-functioning of the male, that is anther part of the flower, that is not proper development of microsporogenesis and microspore tetrad, which ultimately reduce or will ultimately defect the growth of the growth or formation of the gamete within the male part. So you can see here the effect of T toxins, Colicobolus heterostrophus in TCMS maize. TCMS maize variety are affected with this toxin. This toxin, as it attack over the mitochondrial membrane. Here in this picture you can see this toxin produce a different number of pores. With the help of this toxin, this mitochondrial membrane get apart, or due to this toxins, the membrane, the mitochondrial protein membrane, mm, create a pores on the membranes and causes leakage of small molecules and subsequently inhibit APT synthesis and resulting the cell death. Next is the other cultivars are resistance to the pathogen and the toxin. T toxin is essentially for the pathogenicity because it have a virulence nature. T toxin is a mixture of long linear polyketone derivatives. Poly means number of monomers, ketones mean ketone group, that is C double bond O. Then the toxins specifically affect the selective permeability of the mitochondrial membrane, inhibits ATP synthesis, as I have already discussed in the last diagram how the toxins create a pores in the membrane of the in the mitochondrial membrane and causes leakage of small molecules and subsequently inhibit ATP synthesis. Toxins is specific to receptor. The receptor of this toxin is URF13 present in the intermitochondrial membrane of the host cell. Then the next is the HC toxin. The HC toxins produced by the Coccilobolus hemisporium. This pathogen causes leaf spot disease in maize, and this SC toxin is toxin is specific for maize varieties or maize plant. Specific to specific cultivar, detailed mechanism of action of SC toxin is not understood, but the biochemical and other molecular genetic basis of resistance against SC toxin is understood. The resistant varieties of cones have gene HM1 coding for an enzyme toxin reductase which reduce and detoxify the toxins, and this particular gene is absent in susceptible varieties of cones. And those varieties don't have this HM1 gene, so these varieties are susceptible to this toxin. Otherwise, the enzyme toxin reductase are responsible for the detoxifying of this toxin. Last is the host specific toxin, AM toxin produced by alternata, that is this pathogen causes alternaria leaf blotch of apple. This toxin is particularly is for apple variety. AM toxin is a cyclic peptide and usually existing as a mixture of different forms. AM toxin acts selectively on susceptible varieties of apple, as it is coming under host specific, so it is selectively susceptible, act on selectively susceptible varieties of apple. Resistant varieties tolerate AM toxins even more than 1000x concentration. As we know, those are the resistant varieties. That this means they are having a ability to restrict the attack of the toxins. Even if the concentration of toxin is more than 100,000x, even then resistant varieties can tolerate the effect of the AM toxin. The AM toxin causes invagination. Invagination means to get entered by piercing or through membrane of the plasma membrane in a susceptible host. The toxin also causes rapid loss of chlorophyll, that is chlorosis, that is yellowing of leaf. This is a disease also caused by this AM toxin. And other toxin or plant pathogen interaction, you can see here in this table. Some toxin of fumic acid are caused by rhizopus species, and category is fungi. Then diseases almond hull root, alternaric acid, fusoxin, pyrrolidin, then ceratolumin, lycomycin, mycin, then coronatin, teg- Teget toxin, thexomins. These are the number of toxins produced by different type of pathogens, and their category is also mentioned, and even the diseases are also mentioned in this table. You can see how these toxins are, in what type of host they have for their attack. With this, thank you very much.